Right, so let's give this a whirl. So we are going to have a look at how glucose and amino acids are going to be absorbed from the lumen of the ileum and then into the capillaries. So if we start to look at the ileum itself, so remember the ileum is the small intestine. The innermost tissue layer, the epithelium, is highly folded, and each one of these folds, if we have a look at them, have a look at it in a bit more detail, that is one villus. And the advantage of the epithelium being highly folded is it increases the surface area, so more absorption by diffusion prominently can take place. So this is one epithelial cell, and if we have a look at one of these epithelial cells in a bit more detail, we can see that the membrane which faces into the lumen itself is highly folded, and we refer to that as microvilli. Again, highly folded membrane which increases the surface area, so more diffusion predominantly can take place. So that's one epithelial cell. Just underneath, or just below rather, the epithelium itself, we know that we've got a capillary network, and it's into the capillary network, into the capillaries, that the glucose and the amino acids are going to be absorbed into. So we need to have a look at the process of absorption in more detail. So we're going to have a look at the different intrinsic transport proteins that are involved. It uses three different types. One is a carrier protein, which is called a sodium-potassium pump. We have two types of channel protein. And facing in towards the lumen itself, we have another type of channel protein. So if I just draw the epithelial cell, so again, this is the epithelial cell, this is the lumen of the ileum, and we're interested in looking at how glucose and amino acids are going to be transported from the lumen and then ultimately through the epithelial cell and into the capillary. Right, the whole process is going to start off with this carrier protein, which is called a sodium ion, potassium ion pump. It's going to do active transport. We also need to have a look at and remind ourselves that within the lumen we've got amino acids. So if I put a little key on the side over here, those are amino acids. In the lumen there are also glucose molecules. It's my attempt at drawing some hexagons. So glucose molecules. And there are also sodium ions within the lumen as well. Okay. So as I said, the whole process is going to start off with a sodium-potassium pump, so a type of karyoprotein. We'll look at sodium-potassium pumps and a couple of other topics in biology as well. But sodium-potassium pumps are slightly unusual because they transport both sodium and potassium ions, but in particular directions. The sodium-potassium pump will always actively transport sodium ions from inside the cell to outside the cell. So in this case, sodium ions are going to be actively transported from the epithelial cell and then into the capillaries just below them. Potassium ions are actively transported, always in the other direction, so from outside the cell to inside, but they play no role in the reabsorption of glucose or amino acids, so we don't need to mention anything more about them, but that's the direction in which they are transported. The significance of this is that the concentration of sodium ions inside the epithelial cells is now relatively quite low in comparison to the concentration within the lumen of the ileum. And so what's happened is a concentration gradient has been set up and established. What happens now then is that those sodium ions in the lumen now start to move down their concentration gradient through this type of channel protein, which is called a co-transport protein. Co-transport because what's going to happen is this: as the sodium ions move down their concentration gradient, some amino acids will bind with them. But it's the sodium ions that are moving down their gradient, but amino acids will bind with the sodium ions as they move through the co-transport protein. And all we need to say is we can use the phrase that the sodium ions carry the amino acids through the co-transport protein and then into the epithelial cell. And as soon as the sodium ions move through into the epithelial cell, the amino acids are released and they separate. But the effect is that amino acids are transported through into the epithelial cell. Similarly, as more sodium ions move down their concentration gradient through the co-transport protein, some of them will bind with glucose molecules in the lumen of the ileum, and the sodium ions will carry the glucose molecules through into the ileum, and as the sodium ions move through, the glucose molecules will be released and they separate, but the end result is that glucose molecules have been transported through into the ileum from the lumen. And this is referred to as co-transport. What we have now then is a relatively high concentration of amino acids in the epithelium. There's a higher concentration of amino acids in the epithelium compared to the capillary. The capillary is transporting blood, it's constantly flowing, and so it helps to maintain a concentration gradient between the epithelium and the blood. And so amino acids can just move down a concentration gradient through channel proteins. These are just channel proteins. so channel protein. The amino acids move down their concentration gradient by facilitated diffusion. The amino acids have been absorbed into the blood. Similarly, glucose is at a relatively high concentration in the epithelial cell compared to the capillary, and they can move down their concentration gradient through channel proteins and then into the blood. This process just continues. The sodium ions are constantly moving down their concentration gradient. The sodium ions inside the epithelial cell are constantly being actively transported out, maintaining that gradient by the sodium-potassium pump. So ultimately, the end goal has been achieved. Amino acids and glucose have been reabsorbed back into the blood within the ileum.